Hi student, this is Professor Harmony Kim, and this video is the lecture of nervous system and nervous tissue. Let's imagine that a hungry man prepares to drink a cup of hot soup. He smells the aroma and he quickly raises the cup to his lips and tongue. Then suddenly he jerks a cup away from his lips because the soup is so hot that it burns his lips and tongue. None of these sensations, thoughts, emotions, and movement would be possible without the nervous system. Nervous system is responsible for sensation, mental activity, and control of the muscles and many glands. So the nervous system is involved in many body functions. It is the master control and communication system of the body. The nervous system continuously monitors and processes sensory information both from the external environment and from inside your body. What could be the function of a nervous system? Sensory input. So sensory receptors monitor numerous external and internal stimuli. Some stimuli result in sensation you already know, such as light, vision, hearing, touch, body position, and temperature. Other stimuli, such as pH or blood pressure, are processed at an unconscious level. So those informations can be gathered together. And another function of the nervous system is integration. Here, brain and spinal cord are the major organs for processing sensory input and initiating responses. The sensory input may produce an immediate response, or maybe sensory input may be stored as memory and taken and retrieved later, or some sensory input may be ignored. Motor output. Skeletal muscles normally contract only when stimulated by the nervous system. Therefore, through the control of skeletal muscle, the nervous system controls the major movement of the body. The nervous system also participates in controlling cardiac and smooth muscle and many glands. Homeostasis. The nervous system plays an important role in the maintenance of homeostasis. This function depends on the ability of the nervous system to detect, interpret, and respond to change in internal and external conditions. In response, the nervous system can stimulate or inhibit the activities of other systems to help maintain a constant, stable internal environment. In this figure, this person decides to drink water because he feels thirsty. You sense thirsty and you think in the brain, oh, I need to drink water. I feel lack of water in my body. Then you decide your motor output, grab cup of water and start to drink. This nervous system controls to maintain homeostasis, such as stable internal environment. Last function of nervous system is mental activity. The brain is the center of mental activity, including consciousness, thinking, memory, and emotion. So this mental activity is more close to the field of psychology. And also, biology also includes mental activity. But in this course, we will more focus on sensory input, integration, motor output, and a little bit homeostasis. The nervous system is made up of brain, spinal cord, nerves, and sensory receptors, and it can be divided into central nervous system and peripheral nervous system. The central nervous system is the integrating and command center. It consists of brain and spinal cord. If you look at this figure, the yellow color, brain and spinal cord, yellow color indicates central nervous system. Central nervous system integrate, store, and respond to information from the peripheral nervous system. Peripheral nervous system consists of nerves extending from brain called cranial nerve. There are 12 pairs of cranial nerve, and then you study 6 pairs of cranial nerve in the lab. And spinal nerve extend from spinal cord. The peripheral nervous system link all regions of the body to the central nervous system. Therefore, peripheral nervous system detect stimuli, transmit information to and from the central nervous system. So you can see this red color represent peripheral nervous system. And in this figure, there's no cranial nerve appear, but you can see spinal nerve extend from spinal cord. The ganglia is the cell body that lie along the nerve in the peripheral nervous system. We will see ganglia later. Let's think about sensory and motor pathway. And sensory receptors are the ending of nerve cells, or sensory receptors can be separate specialized cell which can detect pain, touch, pressure, light, sound, and other stimuli. Do you remember Merkel cell in the stratum basal of the skin? Or photoreceptor in the eye, or hair cell in the inner ear? These are the special sensory receptor. They are distinctive structural cell which has capacity to detect sensory information. Sensory signals picked up by sensory receptors are transmitted to the peripheral nervous system, the central nervous system. Central nervous system in the brain is the major site for processing information, initiating responses, integrating mental processes, and generate response. The motor division of the peripheral nervous system conduct nerve impulse from central nervous system to muscle and gland. The sensory input is referred to as afferent division. This afferent division transmits action potential to central nervous system from sensory receptor. The motor division, in other words, efferent division, transmit action potential from central nervous system to effector organs such as muscles and glands. So sensory division can be called afferent. Motor division can be called efferent pathway. So it's just one letter is different, so you can maybe memorize S-A-M-E, same. So sensory input is referred to as afferent pathway, afferent division. Motor output can be referred to as efferent output, efferent pathway, efferent division. This figure is from the text, and here's central nervous system. The peripheral nervous system has two divisions. One is sensory division, and the other one is motor division. Sensory division, we just saw that afferent division. Afferent division transmit action potential from sensory receptor to peripheral nervous system, eventually to central nervous system. Now, central nervous system integrate sensory information and send action potential to effector organ, and it could be muscle or glands. This motor division, efferent division, can be divided further. One is somatic nervous system, the other is autonomic nervous system. And this autonomic nervous system further subdivide into sympathetic and parasympathetic division, and sympathetic division most active during physical activity, and parasympathetic division is regulating resting function and control the digestive system. This diagram illustrates the functional organization of the peripheral nervous system. I mentioned that peripheral nervous system covers broad region of the body. Therefore, peripheral nervous system further divided into several divisions. First, sensory division, afferent division, and motor division, efferent division. Again, sensory, afferent, S-A, motor, efferent, M-E, same, S-A-M-E. Let's look at a sensory afferent division first. Afferent division further divided into somatic sensory and visceral sensory subdivision. When sensory receptor innervated in visceral cavity, we call them visceral sensory division. The sensory receptor innervated rest of body except visceral cavity. We call them somatic sensory subdivision. Do you remember when you studied body cavity? Here is visceral cavity. If sensory receptor innervated in visceral cavity, we call that visceral sensory subdivision. If sensory receptor innervated the rest part of body region, we call them somatic sensory subdivision. For example, somatic sensory subdivision can be the sensation of touch, pain, pressure, temperature, proprioception, body work, and limb. And this general and special means, general means sensory receptor distribute widely, broadly, then that is general. Special means a sensory receptor distribute in limited area, especially in head. Then we call them special sensory subdivision. For example, hearing and equilibrium by ear, vision by eye, and smell by nose. These are the special somatic sensory subdivision. Sensory receptor innervated in visceral cavity. We call those sensory information visceral sensory subdivision. Also, it divides into general and special. In terms of general, stretch, pain, temperature, irritation in viscera, nausea, and hunger. What about the special? You can have a taste by taste bud. Taste located in the head. Then why it is not belong to special somatic sensory division? Because taste develop in association with the gastrointestinal tract, which is visceral region. Therefore, taste put into visceral sensory category rather than somatic sensory category. Some scientists even put this smell into visceral sensory category because once you have a smell, that stimulates appetite. Therefore, smell also put into special visceral sensory category as like taste. But some scientists, that is indirect way. Once you smell, it goes up to your brain and then based on memory, oh, I remember this smell, I like to eat this food. So a little bit process and then that stimulate your appetite. But taste is directly related to gastrointestinal tract. So there's a little bit disagreement, but taste is definitely belong to visceral sensory subdivision. Once this sensory information transmit to PNS and eventually go up to central nervous system. And from central nervous system send back actions potential to effector organ. So when you look at motor division, efferent division also divided into somatic motor subdivision and visceral motor subdivision. Same way, somatic motor subdivision is motor neuron innervated in body region except visceral cavity. Visceral motor subdivision is the area where motor neuron, involuntary motor neuron, innervate in the organs located in visceral cavity. So one more time, this sensory subdivision, afferent subdivision of the peripheral nervous system is subdivided into somatic sensory and visceral sensory subdivision. And somatic sensory division include general somatic sense, special somatic sense, and proprioceptive sense means give body sense of position and movement of body in space. So proprioceptive sense is have a sensory information of body position in relation to space. Where are you now? At home, maybe, or in a workplace. Every moment you do not sensitize where you are, but suddenly when you think about look around and then look at space, oh, where am I? Or maybe you fell down and then you lose consciousness, and then when you wake up, you do not know and look around, where am I? And then you realize, oh, this is in the hospital or my bedroom or in the living room. This kind of sensory information can be detected by proprioceptive senses. So, somatic sensory subdivision include general somatic sense, proprioceptive sense, and spatial somatic sense. Spatial somatic sense can be this sensory receptor limit in location of a head. We call them spatial somatic sensory. In terms of spatial visceral sensory is definitely taste, because this taste directly related to gastrointestinal organ which are located in visceral cavity. Efferent subdivision of peripheral nervous system also divided into somatic motor and visceral motor. When motor neuron innervate in broad body region, we call that somatic motor subdivision. When motor neuron innervate in visceral region, then we call them visceral motor subdivision. And usually, somatic motor subdivision is controlled by voluntary motor neuron, and visceral motor subdivision is regulated by involuntary motor neuron. This chapter is for nervous tissue. So nervous tissue means similar kind of cells grouped together and have a similar function. So let's look at the cells of the nervous system. The nervous system is made up of neuron and non-neuronal cells called glial cells. So from now on, you will study neurons and types of neuron and glial cells in the central nervous system, glial cells in the peripheral nervous system. Also, you will study myelinated and unmyelinated axons. So these are the cells can be found in nervous tissue. We will study each cell one by one. Neuron first. The basic structural unit of a nervous system is a neuron. Neurons receive stimuli and transmit electrical signal along their plasma membrane. Electrical signal. This is the way how nervous system communicate with other organ system. In terms of a nervous system, they send electrical signal. Electrical signal means nerve signal, nerve impulse, action potential. Those are all same meaning. Nervous system send nerve impulse to adjacent nerve cell or send electrical signal to muscle or to other organ. Neuron cannot divide. Once neuron died, we cannot replace the lost neuron. Neuron has a long lifespan. Neuron can live over 100 years. So it's very important to not lose neurons. As long as a neuron survive, they do not die in short time. But once you lose a neuron, it's impossible to make new neuron. That's why Alzheimer's disease is dangerous, because the patients keep losing, because the nerve cell keep dying. Therefore, human cannot have a normal mental activity. Neuron has a high metabolic rate. The entire mass of neuron is about 2% out of entire body. but it consumes 20% energy which your body can spend. So it carries high metabolic rate. Let's look at the structure of a neuron. Neuron has a cell body, this star-like structure, cell body, and there's a long extension, it's called axon. This cell body is named soma. And if you look at inside the cell body, you can see nucleus and also endoplasmic reticulum, Golgi body, mitochondria, and there's Nissl body or chromatophilic substance. This Nissl body is an aggregate of a rough endoplasmic reticulum and free ribosome. What's going on in the ribosome? Ribosome is the site for protein synthesis. So in this Nissl body, protein synthesis takes place, and Nissl body can be found in the cell body and even in the dendrite. Also, in the cell body, you can find neurofibril. Neurofibril bundles with intermediate filament to form a network between chromatophilic bodies. And when you study nervous system, you will see this terminology. Nuclei, usually you think that many nucleus. In cell biology, when you see the word nuclei in nervous system, it means cluster of cell body in the central nervous system. So this cell body can cluster another neuron, and then they get together, so cell body can be clustered. We call that cluster of cell body in central nervous system is referred to as nuclei. And also, when neurons group them, a bundle of neuron process in the central nervous system, we call that tract. So it kinds of a bundle of axons. We call them tract. But this is the story in central nervous system. In peripheral nervous system, the cell body lie along the nerve. We call that ganglia. And the axonal part in the peripheral nervous system, we call them nerve. When you look at neuron, there are two types of processes. One is dendrite, the other one is axon. Dendrite, dendro means a tree, so look like tree branch. It's short, branched, cytoplasmic extension of the cell body is called dendrite. And many dendritic surface have a small extension called a dendritic spine. So if you look at this dendrite, also you can see small branches coming out like this. We call them dendritic spine. All organelles found in cell body also found in the dendrite. Main function of dendrite is to transmit electrical signal toward the cell body. So you can imagine that some other neurons have interaction with this dendrite. So this dendrite receive information from other neuron and send that information to cell body. So transmit electrical signals toward the cell body. That is the main function of a dendrite. Therefore, dendrite act as receptive site, receiving signals from other neurons. The other process is axon. It's long cytoplasmic extension from the cell body. It is called nerve fiber because it is long, because of their shape. Transmit nerve impulse away from the cell body. This time dendrite transmit information to cell body. Now cell body send nerve impulse away from the cell body, which is through axon. So transmit impulse away from the cell body and generate action potential to other cell. When you look at the end of axon, this axon branch at the end of an axon, we call this axon terminal. Axon terminal store neurotransmitter chemicals in tiny vesicles. So this is axon terminal. Then axon terminal is usually a little bit bulge. This inside the axon terminal there's numerous vesicles, and inside the vesicles there are neurotransmitter. Neurotransmitter, chemical messenger molecule that excite or inhibit adjacent neuron. Throughout axon, neurofilament, actin microfilament, and microtubule, this kind of cytoskeleton provide structural strength along the length of axon. There are three types of neurons based on the extension. One is a multipolar neuron. Multipolar neuron has several dendrites and a single axon. Most 99% of neurons belong to multipolar neurons. Usually interneuron and motor neurons are multipolar neurons. Motor neurons innervate in effector organ in peripheral nervous system. Purkinje cell of cerebellum or pyramidal cell shows multipolar neuron. Pyramidal cells are found in cerebral cortex, hippocampus, and amygdala in the brain. Bipolar neurons have two processes extending from opposite side of the body. So here's the cell body. Usually dendrite located close to cell body, but in bipolar neuron, dendrite can be formed further from cell body and look like two processes extending from opposite side of the body. Bipolar neuron can be found in olfactory cell and retina cell. So here's retina. Retina has 7 different kinds of cells, and one of them is bipolar neuron. And also olfactory neuron belong to bipolar neuron. This is unipolar neuron. When you look at unipolar neuron, here's the cell body and then a little bit extension, very short extension. Then this extension divide into two, look like inverted T, and this extension is a long branch. So one side is called peripheral process, the other side is called central process. Central process carries axon terminal. Actually, the formation of unipolar neuron starts from bipolar neuron. This is a bipolar neuron. And then from this bipolar neuron, a little bit extension, very short extension. From this extension, then it branches into opposite side. Therefore, unipolar neuron start as bipolar neuron during development. That's why this neuron has pseudo-unipolar. It begins from bipolar. Therefore, it is not genuine unipolar. So it has another name, pseudo, means fake pseudo-unipolar neuron. Or we can simply call unipolar neuron. So which one is dendrite, which one is axon? This is definitely axon because it has axon terminal. But what about this? Can we call this as dendrite? Actually, no. Peripheral process is functionally similar to dendrite. However, the peripheral process is called an axon because its structure is identical to that of true axon. Therefore, unipolar neurons do not have dendrite. At the terminal end of the peripheral axon, receive sensory stimuli and transmit a signal through the peripheral axon toward the cell body. So still this peripheral process receives sensory stimuli, but we do not call this part dendrite because the structure really similar to axon, so we call this peripheral process. And this part we call central process. So there's no dendrite in unipolar neuron. Where you can find unipolar neuron? In dorsal root ganglion cell. This is spinal cord. Hope you remember this is anterior horn, this is posterior horn. In posterior horn, there's sensory neuron. Look at this sensory neuron. Here's cell body and then two long extension. It shows unipolar neuron. This sensory neuron receive sensory information and transmit to spinal cord. So when you see here's cell body and here's axon, And then the other end of a peripheral process look like dendrite, but we do not call this dendrite. We just call the end of peripheral process. And then this end of peripheral process act as recessive ending. I mentioned that in nerve tissue, there are other cells in addition to neurons. We call them glial cells. Glial cells are non-neural cells that support and aid the neurons of the CNS and PNS, provide supportive scaffolding for neurons, and insulate the non-synaptic part of a neuron. The number of glial cells is much more than neuron. The ratio between glial cells and neuron is about 10-to-1 ratio, make up half the mass of the brain. If there's a brain disorder occur, there are more probability to the problem on the glial cell rather than on the neuron. Glial cells also have a central body and branching processes. Glial cells divide throughout life. They have a capacity to undergo cell division, so they can reproduce themselves. 6 kinds of glial cells found in nerve tissue: astrocyte, ependymal cell, microglia, oligodendrocyte, satellite cell, and Schwann cell. Look at this microscopic view. So here's cell body, and here's another cell body, and then maybe backward of this screen there should be axon. But when you look at this purple dot, they are all glial cells. So think about the number. You can see only 1 or 2 neurons in this view, but there are so many glial cells. So the population of glial cells is much higher than that of neurons. Which dot indicate which glial cell we cannot recognize in this figure. We need more higher magnification. But you can. Let's look at the glial cells in the central nervous system first. Astrocyte, ependymal cell, microglia, oligodendrocyte. These are the glial cells found in the central nervous system. Here's astrocyte. There's no ependymal cell in this figure, but here's microglia and oligodendrocyte. We will look at each glial cell one by one. First, astrocyte, most abundant glial cell in nervous tissue. What does astrocyte do? Astrocyte recapture and recycle neurotransmitter. So, astrocyte regulate neurotransmitter levels by increasing the rate of neurotransmitter uptake in the regions of high neuronal activity. Also, signaling increased blood flow through capillaries in active region of the brain. So, astrocyte signal increase blood flow through capillaries, especially in active region of the brain. Also, astrocytes take up and release ions to control the environment around the neurons. Astrocytes control the ionic environment around the neuron, so that can influence the release of neurotransmitter. Also, astrocytes help synapse form in developing neural tissue. Astrocytes produce molecules necessary for neural growth, propagate calcium signals that may be involved in memory. It looks like star shape. Astro means star-like, so it shows star-shaped glial cell. Microglial cell. This is a microglial cell, smallest and least abundant glial cell in central nervous system. Phagocytized microorganism, foreign substance, and necrotic tissue. Microglial cell derive from blood cell called monocyte. So there are several cell types in blood, and one of them is monocyte. This monocyte creeping out of the blood vessel and stay in the interstitial space in tissue. Once monocyte in the blood vessel creeping out of the blood vessel and stay in interstitial space of tissue, we call them macrophage. Especially when macrophage present in the central nervous system, we call them macroglial cell. And why they creeping out of the blood vessel? Because they wanna grab microorganism, foreign invader, and even some dead tissue. They phagocytize all this stuff. Another glial cell is ependymal cell. Ependymal cell, the wrapping garment, form a simple epithelium that line the ventricles and the central canal of the spinal cord. So you can see here, lining ventricles. What is ventricles? Very narrow space, narrow chamber inside the brain. Some ependymal cells are specialized to produce cerebral spinal fluid. We call them CSF. In other words, inside the brain, there is some fluid flowing. So brain is kind of immersed in the fluid. It's kind of suspended in fluid. Ependymal cell has cilia, as you see here, cilia. These cilia to help circulate this CSF. A ventricle is not wide open space. It's a very narrow, tiny chamber. Therefore, these cilia of ependymal cell facilitate circulating cerebrospinal fluid. Let's look at a little bit detail about cerebrospinal fluid. Ependymal cell and associated blood vessels that produce cerebrospinal fluid are called choroid plexus. Choroid plexus is the region in which cerebrospinal fluid can be produced. And in that region of choroid plexus, ependymal cell line up. So here's ependymal cell and here's blood vessel. We call this region choroid plexus. In this choroid plexus, cerebrospinal fluid can produce. So 80 to 90% of cerebrospinal fluid is produced by choroid plexus within lateral ventricle. So the lateral ventricle at the center in the brain, that is the region choroid plexus located. That is the reason CSF can be produced. About 10 to 20% of cerebral spinal fluid produced in the third and fourth ventricle. So brain float rather than being tightly attached to the skull. This CSF fluid provides cushion. Therefore, when your brain hit, fluid kind of provide cushion and protect the brain. Another function of CSF is buoyancy. Because the brain is immersed in fluid, the weight of brain is reduced from 1,400 grams to 50 grams. Imagine that if you hold 1.4 kilogram every moment, at the beginning it might not be that heavy, but you have to hold 1.4 kilogram of brain on the top of narrow neck. You must feel heavy at the end of the day. Thanks to this cerebrospinal fluid, that make you feel reduction of weight. So you hold your brain like a 50 gram of brain on the top of neck. And this cerebrospinal fluid can be produced in the region of choroid plexus. This choroid plexus consists of ependymal cell, one of glial cell in central nervous system. So let's look at here. Cerebrospinal fluid produced by ependymal cell. So here's lateral ventricle. I'm sorry, this is not later. This should be lateral ventricle. Typo. Lateral ventricle here. And this lateral ventricle is composed of choroid plexus. And this choroid plexus, you can see this region, choroid plexus, is composed of ependymal cell and blood vessel. Once CSF produced, this CSF release from lateral ventricle into third ventricle. This is a third ventricle. And then start to circulate from lateral ventricle, third ventricle, and cerebral aqueduct. And here there's a notch in cerebellum. We call that fourth ventricle. Keep circulating from fourth ventricle in spinal cord. Do you remember central canal at the center hole of spinal cord? That is the space for CSF circulate. Once it finishes circulating spinal cord, it comes back and then it enters subarachnoid space. Subarachnoid space is, do you remember, brain skull has dura matter, arachnoid matter, and here's pia matter. Pale purple line indicate arachnoid matter. And this is pia matter. And this is arachnoid matter. Between pia matter, this is pia matter, and this is arachnoid matter. The space between pia matter and arachnoid matter, we call that subarachnoid space. So this subarachnoid space, CSF circulate. Then here's some sinus. Then through this sinus, CSF can drain into blood vessel. CSF drain into the interjugular vein, eventually enter the vein of the general circulation. And then in the middle of circulation, throughout brain and spinal cord, some excretion of waste product can be collected to CSF and then eventually try to get rid of this waste from the brain. So every day human produce 300 to 500 milliliter of CSF generate. And glial cell involved in production of CSF is ependymal cell. The last glial cell in the CNS is oligodendrocyte. Oligodendrocyte, few branched cell, have a few branches than astrocyte, but still have some branches. One of the characteristics of oligodendrocyte is wrap their cell processes around thicker axon. So this yellow line indicates axon. Well, we call them nerve fiber, same meaning. And oligodendrocyte produce myelin. This myelin surround and wrap around the axon, so we call that myelin sheath. So in the lab, you hear about Schwann cell. Schwann cell is forming myelin sheath. That is the story in peripheral nervous system. In central nervous system, it is oligodendrocyte which produce myelin and form myelin sheath. Since myelin sheath wrap around the axon, when you see thicker axon, that thicker axon implies myelinated axon. There's unmyelinated axon. Those axons are thin axon. What about the glial cells in the peripheral nervous system? Satellite cell and Schwann cells belong to glial cells in the peripheral nervous system. Let's look at satellite cell first. This is a satellite cell. It provides support and nutrition to the cell body of a neuron. So it nourishes neuron cell body within ganglia. Protect neuron from heavy metal poison such as lead and mercury by absorbing them and reducing their access to the neuron cell body. So you can see satellite cell found in peripheral nervous system. Schwann cell. Schwann cell, another name is neurolemmocyte, form a myelin sheath around all axons in the peripheral nervous system. What is myelin sheath? Myelin is the lipoprotein. Lipoprotein means phospholipid associated with protein. It's like a membrane. Phospholipid bilayer. In the middle of a phospholipid bilayer, membrane protein innervated. So, myelin is a lipoprotein because of these phospholipids like fat. Therefore, myelin sheath shows white in color. Myelin is formed by Schwann cell, which is wrapped in concentric layer around the axon. It creates a tightly packed coil of membranes. During the wrapping process, the cytoplasm is squeezed from between adjacent layer of Schwann cell membrane, so a tight core of plasma membrane material encompasses the axon. This wrapping is called myelin sheath. The Schwann cell nucleus and a bulk of cytoplasm end up just beneath the outermost portion of its plasma membrane. The peripheral part of Schwann cell and its exposed plasma membrane, the outermost of plasma membrane, is called neurolemma. Since this myelin sheath is formed by many individual Schwann cells, so Schwann cell in this myelin sheath, another Schwann cell in this myelin sheath. Many individual Schwann cells form myelin sheath. Therefore, there's this continuous sheath, like this gap. So this continuous sheath separated by gap, we call this node of Ranvier. Node of Ranvier. There is a bare area of axon in length which is not covered with myelin. Unmyelinated axon rests in invagination of the Schwann cell, but the Schwann cell's plasma membrane does not wrap around it many times. So it shows thin axon. Unmyelinated axon is a thin axon. Myelinated axon is thick axon. What does myelin sheath do? Electrically insulate axons from one another. What is the component of this myelin sheath? Lipoprotein. It's fat. Then the fat insulate electrical current. This is a microscopic view of a myelin sheath. So this is a bundle of axon, like this. Here's a bundle of axon. Here's single axon surrounded by myelin sheath. There's many myelinated axon. Bundle them together. And imagine that you cut longitudinally and look at the section side. So here's axon. And this axon wrapped around by myelin. So this part, myelin. And when you look at along the axon, there is some gap, a little bit indented. And here's line. We call this node of Ranvier. This glial cell found in central nervous system. In contrast to Schwann cell, each oligodendrocyte have multiple processes that coil around several different axons. So here's oligodendrocyte. Schwann cell, individual Schwann cell wrap one axon, but oligodendrocyte has multiple process like this here and here, multiple process. Therefore, each process can coil around several different axons. That is the difference between oligodendrocyte and Schwann cell. Schwann cell wrap around one axon, but oligodendrocyte can wrap multiple axons, several different axons. What is the significance of myelin sheath produced by oligodendrocyte or Schwann cell? Let's think about electrical signal. Electrical signal is the means to communicate with other organ system. So neurons conduct electrical signal along the length of their plasma membrane. Here's axon, and here's axon, and in resting neuron, unstimulated neuron, the membrane is polarized. So cytoplasmic side is negatively charged with respect to the extracellular side. This is the portion of the cell of axon. And inside the axon, inside the nerve cell, is negatively charged because of phosphate of DNA and some protein, negatively charged protein. Therefore, inside the cell is negatively charged in resting state. And outside the cell is positively charged because there are more sodium ions than inside. Therefore, outside the cell, extracellular matrix is positively charged. Inside the cell is negatively charged. Therefore, we call this polarized state. Polar means South Pole, North Pole. It's kind of two different extremes. We call that polarity, polarized. So inside the cell is negative. Outside the cell is positively charged. So they have opposite charges. Therefore, we call that polarized. When a neuron is stimulated, the permeability of plasma membrane changes at the site of the stimulus, allowing positive ions to rush in the cell. As a result, the inner face of the membrane becomes less negative. This positive ion can enter the cell. How? There is some ion channel protein in the plasma membrane. This is transport membrane protein. Control the ion across the membrane. Ion is very hard to freely enter the cell because the hydrophobic region of a phospholipid dislike ion. But there is some channel protein. This channel protein open, then sodium ion can easily enter the cell. When nerve impulse arrive in this region, then ion channel protein in the plasma membrane open, allow sodium ion enter the cell. So this portion locally depolarized. Now, inside the cell become positively charged, outside the cell also positively charged. So it's not much different. We call that D, not polarized. Local current make this region of the cell depolarized. Once this depolarized occur, then this nerve impulse continue to stimulate adjacent portion. Therefore, there's another ion channel protein in the plasma membrane. This also open again. Therefore, positive ion such as sodium ion can enter. So now this region become depolarized. Action potential is going to propagate according to the ion channel protein in plasma membrane. This is the case of non-myelinated axon. But think about that. This action potential need to propagate one by one, and it takes long time. This action potential eventually arrive at the axon terminal. Therefore, this nerve impulse need to transmit to the next neuron. Until that time, it takes so long. But if axon has myelin sheath, this myelin sheath insulate electrical current, but there's a gap. Here's nodal Ranvier. Only this gap region, ion channel protein can be exposed. Therefore, electrical current doesn't have to propagate one by one in small scale. Action potential can propagate like this: jump, jump, jump. This speed up to transmit action potential to the next neuron much faster than unmyelinated axon. So, myelin sheaths facilitate propagating action potential along the axon as fast as possible. Synapse. Synapse means the junction between two cells. Synapse means the site at which neurons communicate with each other. So signals pass across synapse in one direction. It goes one direction. It does not go back and forth, not that way. Based on synapse, the cell that transmits a signal toward the synapse, we call that presynaptic cell. So this is a presynaptic cell. Why? Because a presynaptic neuron sends signal toward the synapse. This cell is called postsynaptic neuron. Why? The cell received a signal, which is called postsynaptic cell. Postsynaptic cells can be either neuron or can be muscle or gland. So do you remember neuromuscular junction? And motor neuron, and then this can be muscle fiber. And the neuromuscular junction is the synapse, junction between motor neuron and muscle fiber. Usually, synapse can be notified this cell body, axon terminal, cell body of another neuron. So this is presynaptic neuron, and this is postsynaptic neuron, cell body, axon terminal. In between two neurons, synapse is formed. There are several types of synapse. Axodendritic synapse, axosomatic synapse, axo-axon synapse, dendrodendritic synapse, dendrosomatic synapse. Let's look at axodendritic synapse first. Just to look at the word. Axo means axon of a presynaptic neuron, synapse with dendrite of postsynaptic neuron. So between axon terminal of one neuron and dendrite of another most common type of synapse. For example, this one, axodendritic. Blue color region is axon of presynaptic neuron. So axon terminal, synapse with postsynaptic neuron, and here's a dendrite. So axon terminal of a presynaptic neuron, synapse with dendrite of a postsynaptic neuron. We call that axodendritic synapse. Axosomatic. Here's axon terminal of presynaptic neuron, synapse with cell body of postsynaptic neuron. We call that axosomatic synapse. Axonic synapse. Here's axon terminal of presynaptic neuron, synapse with the beginning part of axon of postsynaptic neuron. Dendrodendritic and dendrosomatic synapse are very rare case, and these are not shown in this figure. What's going on in the synapse? Presynaptic terminal are specialized to produce and release neurotransmitter. This is axon terminal of presynaptic neuron, and this is postsynaptic neuron, or maybe some other specific cell such as muscle fiber. The major cytoplasmic organelle within presynaptic terminal are mitochondria and numerous membrane-bound synaptic vesicles, and these synaptic vesicles contain neurotransmitter. So when you study neuromuscular junction, what is the name of neurotransmitter found in the vesicle? Acetylcholine. So when you look at axon terminal of a presynaptic neuron, here's mitochondria and several vesicles. Inside the vesicle, neurotransmitter. Upon arrival of action potential at the presynaptic terminal, neurotransmitter are released from the vesicle into the synaptic cleft. There is a narrow space formed between two neurons. We call that synaptic cleft. Once action potential arrives at the end of axon terminal, then neurotransmitter release into synaptic cleft. Neurotransmitter diffuse across synaptic cleft to bind to membrane receptor on the postsynaptic cell. So postsynaptic cell also has receptor which is ready to bind to neurotransmitter. Once neurotransmitter bind to receptor of postsynaptic neuron, then that triggers action potential to postsynaptic neuron. Several substances have been identified as neurotransmitter. Acetylcholine, you already studied, excitatory in central nervous system so that your muscle can contract. The serotonin, GABA, dopamine, glycine, norepinephrine, these are the neurotransmitter. Depending on the characteristic of neurotransmitter, once neurotransmitter release and bind to receptor of postsynaptic neuron, then some neurotransmitter activate postsynaptic neuron. Some neurotransmitter deactivate postsynaptic neuron. For example, serotonin is happy neurotransmitter. So it inhibits depression. In case of a GABA neurotransmitter, it inhibits motor neuron. Dopamine, it controls voluntary movement. If dopamine is not released from presynaptic neuron, or sometimes if too much dopamine release, it prevents your voluntary movement. So neurotransmitter is important. If neurotransmitter does not function normally, then postsynaptic neuron cannot be stimulated. Maybe when you are young, your mom gives you a milk before you sleep. When you drink milk, serotonin can be released, and then this serotonin plays a role in happiness. Therefore, you can have a cozy, happy sleep. So overall, serotonin neurotransmitter mediate happiness, satisfaction, and optimism. So it is the best way to release neurotransmitter in a natural way. If you want to pursue too much happiness, then if you take some drug, this drug can release serotonin neurotransmitter much higher up. So you might feel satisfaction, but next time in order to make yourself satisfied, you need to take more drug that induce addiction. So releasing neurotransmitter should be natural way. How you can induce natural way of neurotransmitter? So the several organ system can well communicate with the nervous system. So this kind of a neurotransmitter can properly release depending on the situation. In the lab, we study white matter and gray matter. Nerve tissue is organized axon and cell body. This axon can form bundle. Also, cell body can form bundle. Once they bundle, they show some color. In terms of axon, it's wrapped around myelin sheath. Therefore, they show white color. We call that region white matter. And the central area, this is a spinal cord. The central area of spinal cord is gray matter. And the outer surface of much of the brain, this outer surface of much of the brain consists of gray matter called cortex. This region is a cortex. So the gray matter of the central nervous system perform integrative function or act as relay area in which axons synapse with the cell body of other neurons. So this gray area is more like cell body region, bundle of cell body region. And interestingly, the central part of spinal cord has gray matter, and outside spinal cord composed of white matter. In contrast, in brain, the center part is white matter. Of course, here is gray matter region, but mostly center part is white matter, and outside region is cortex region is gray matter. So the distribution of gray matter and white matter between spinal cord and brain is opposite. White matter bundles of parallel axons with their associated myelin sheath. Because of a fat component of myelin, therefore, white matter is the region in which bundle of parallel axons are formed. Gray matter, collection of neuron cell body region. Unmyelinated axons, they form gray matter. So when you look at this spinal cord, this is anterior horn, this is posterior horn. Most nerves contain myelinated axon, but some consist of unmyelinated axon. So when you look at gray matter in the spinal cord, here's H-shaped region surrounded central cavity. Dorsal half contain cell body of interneuron. Here, dorsal half contain cell bodies of inner neuron. Can you see? Here's inner neuron. And ventral half contain cell bodies of motor neuron. Here's motor neuron. Cell bodies are clustered in the gray matter. When you look at white matter in spinal cord, white matter located externally to the gray matter and contain no neuronal cell body, but millions of axons, myelin sheath. So white matter in the spinal cord located externally to the gray matter, and white matter contain no neuronal cell body, but millions of axons compose white matter. And just briefly, a study about new terminology in nervous system. So, cluster of cell body in central nervous system is called nuclei. And white matter in central nervous system is called tract. Bundles of axon in central nervous system is called tract. Cell bodies in peripheral nervous system, we call that ganglia. Neuron process, such as axon in peripheral nervous system, we call them nerve. Let's look at the structure of a nerve. Nerve, a collection of axons in the peripheral nervous system. We saw that just before. So the nerve consists of collection of axons, Schwann cells, and connective tissue in the peripheral nervous system. So when you look at this axon wrap around myelin sheath, and each axon wrap around the myelin sheath also covered by connective tissue called endoneurium. So within a nerve, each axon is surrounded by Schwann cell, then a layer of loose connective tissue called endoneurium. Bundle of axons, we call that nerve fascicle. This nerve fascicle, this bundle of axons, surrounded by connective tissue, we call that paraneurium. And entire nerve is surrounded by tough fibrous sheath, we call that epineurium. It's similar to muscle. Muscle fiber is surrounded by endomysium, and bundle of muscle fiber, we call that muscle fascicle. Muscle fascicle surrounded by paramysium, and entire whole muscle is surrounded by epimysium. It's a similar pattern of a muscle structure, but this time the name is endoneurium, and nerve fascicle, and paraneurium, and epineurium. Now let's look at functional aspect of a neuron. Sensory receptor transmits sensory impulse toward central nervous system. And central nervous system send action potential to motor neuron. Let's look at that pathway a little bit detailed. So neurons are grouped functionally according to the direction the nerve impulse travel relative to the central nervous system. Here's afferent transmission. Sensory neuron transmit sensory impulse toward central nervous system. Sensory neurons have a short, single process divided into central process and peripheral process. This is unipolar neuron. All sensory neurons are unipolar neuron. Unipolar neuron has central process. This is axon terminal, and here's peripheral process. We call this site also axon. But at the end of a peripheral process, there's receptive ending. So this receptive ending receive sensory impulse and transmit sensory information toward cell body, continue to central nervous system, in this case spinal cord. Cell body of this unipolar cell is swelling. We call this a ganglion. And this is a dorsal region. Therefore, dorsal region ganglion cell. So ganglion is a structure containing nerve cell body. It forms swelling and link by synapse. Once sensory neuron transmit sensory impulse to central nervous system, it synapses with interneuron. Interneuron lie between sensory neuron and motor neuron, and they are mostly multipolar neuron. These inner neuron form junction with effector cell and then send impulse to motor neuron. And motor neuron also mostly multipolar neuron. Therefore, motor neuron carry impulse away from central nervous system to effector organ. Sensory neuron transmit sensory impulse to central nervous system. We call that afferent transmission. Once interneuron receives sensory impulse from sensory receptor, interneuron synapse with motor neuron and send impulse away from central nervous system to effector organ by way of motor neuron. We call this path efferent transmission. So this is sensory, therefore SA, and this is motor, therefore ME, sensory afferent. Motor neuron efferent transmission. Reflex. What is the meaning of a reflex? It's kind of a response to a stimulus. The characteristic of a reflex is stereotype, unconscious, and involuntary response to a stimulus. A stereotype means the response to the stimulus is always more or less the same. Unconscious means no awareness is necessary for the reflex to occur. It's so fast response. So you might not even have consciously realized, oh, I respond such a way. Involuntary response means one cannot will the reflex to happen. It's sometimes out of control. So reflex take place to maintain homeostasis. Remember, one of the function of the nervous system is maintaining homeostasis. Some reflexes result in the contraction of skeletal muscle like this. You just ouch and you take your muscle away from the candlelight, or a visceral reflex activating smooth muscle, cardiac, or gland. In terms of gland, you can sweat and then you can secrete some oil substance. Reflex are produced by reflex arc. Reflex arc has 5 basic components. What are those 5 components of reflex arc? First, sensory receptor transmit sensory impulse through sensory neuron. And inner neuron receive those impulse and synapse with motor neuron. Transmit action potential to effector organ through motor neuron. So sensory receptor, sensory neuron, inner neuron, motor neuron, and effector organ. These are the 5 components of reflex arc. Most reflex arc have at least one interneuron. This is interneuron. So sensory neuron synapse directly with motor neuron without inner neuron. We call that monosynaptic reflex, simplest of all reflex. Example is knee-jerk reflex. When doctor tap the patella ligament, it stretches quadriceps and excites its muscle spindles. Afferent impulse travel to spinal cord and synapses occur with motor neuron. Look at this red line. This is monosynaptic reflex. Monosynaptic reflex occur, the doctor tap patella ligament. Then it stretches the quadriceps and excites its muscle spindle. This afferent impulse travel to the spinal cord and synapses occur with motor neuron. So look at this. Red line indicates motor neuron. So sensory neuron and motor neuron directly synapse. This motor neuron sends activating impulse to the quadriceps, causing it contract and extending the knee. This is the simplest of all reflex. There is no inner neuron involved. Polysynaptic reflex, then inner neuron is involved. So polysynaptic reflex must have a single inner neuron between sensory and motor neuron. So when you withdraw reflex like this, you touch some sharp pin and then your afferent neuron send sensory information to inner neuron synapse with motor neuron, and then immediately you flex forearm. Therefore, you want to protect from injury. So let's look at anatomical pathway of a reflex. One more time. So this person receive painful stimulus. This painful stimulus trigger nerve impulse in a sensory neuron. Afferent transmit. This initiate polysynaptic withdrawal reflex. And this sensory neuron synapse with inner neuron, and inner neuron send impulse to effector organ, in this case finger, by way of somatic motor neuron. These reflex occur rapidly. But at the same time, once this person has this event, also there's some other pathway also take place. We call that cortical pathway. Then from this inner neuron, somatically, your skeletal muscle detach from painful stimulus. At the same time, some thought arise. This thought process transmit from interneuron through medulla oblongata and arrive at thalamus. This processing simultaneously, the nerve impulse travel on the axon branch that extends into white matter. This ascending axon carries the nerve impulse to the brain. And eventually it arrives at thalamus. And then continue to go up higher, sensory cortex. From sensory cortex, you think that, just before I burn my finger. Then there's another pathway, cortical pathway occur like that, where I think I injured my finger. I think my finger is burned, so I want to repair the injury. Therefore, the thought process, this cortical pathway, then, okay, how can I heal the injury? And this thought process is a non-reflexive motor response is initiated in the gray matter and descending axon and transmit down a descending axon in the white matter to stimulate somatic motor neuron. This time you are going to put finger in the running water of the faucet. This cortical pathway is not involved in reflex arc. So reflex arc begins from spinal pathway such as unconscious and involuntary way. But this event can be followed by cortical pathway, so humans can explore to voluntary response. Okay, this is overall lesson of nervous tissue. Let's solve the question. Question 1. Which of the following is the most abundant cells in the brain? Astrocyte, ependymal cells, microglia, oligodendrocyte? You can find the answer. Question 2. What is it called? The clusters of cell bodies in the central nervous system. Nuclei, tracts, ganglia, or nerves? You can find the answer from the lecture video. This is the example of submitting week 7 assignment. This is exactly the same way you've done weekly base. So you send the email. In the main text body, you write down your last name, comma, followed by first name, and include answers of 2 questions. The due of this assignment is until November 29th, Wednesday midnight. Because next week is Thanksgiving holiday, you will have 1 more week to submit this week 7 assignment. This is the end of lecture video. Thank you.